started then. So I'd like to also thank Kavita and Deepa for this invitation to come. I've had several students and many colleagues from India, so I'm very happy to finally have a chance to visit. And we have a number of collaborative programs, especially for students. So this is partly an invitation for people to visit. This is our institute in Mishima. Right, so Deepa has, in fact, put me in an interesting position because she's given some very nice evidence that selection can act strongly on synonymous mutations, mostly through experimental studies, her evidence was. So what I'll try to present is evidence from population genetic and comparative studies that support weak selection at silent sites. So you can consider it sort of a challenge to decide if you favor one view or another, or if these views can be reconciled. I've been thinking about it for a couple hours. I'm not sure I have an answer. So the motivation for this study really came from basic ideas in evolutionary biology. So an important aspect of Darwin's view of adaptation and the development of complex adaptations, he viewed as the result of basically continuous fine-tuning, right? And so selection discriminating among scarcely appreciable differences. And so Dick Lewontin echoes this view and suggests that evolutionarily significant genetic variation is almost by definition manifest in subtle differences. And this would be in morphology, physiology, behavior between individuals. And so here he states it as an assumption, but this is a testable notion, but one that's difficult to test because experimental studies are limited to finding selection coefficients, usually of the order of 1% or so, where we think that selection in nature might be effective, depending on population size, for many orders of magnitude smaller. Right? And so the suggestion is that selection that may be undetectable in direct measures may be prominent and important in adaptation. And so codon usage then, for me, was a model to try to study weak selection. So I've actually revised the talk, and I started with the wrong one. Just a second here. Version D. Kimura's model of neutral evolution is quite simple. But in contrast, Ota's model, which she referred to as nearly neutral, is a little bit, has some subtleties that make it harder to teach, at least from my perspective. So partly, the term nearly neutral is a little bit deceiving. So the idea is that rather than considering fitness effects of essentially 0, Ota proposed that fitness effects of on the borderline between mutations that would be dominated by drift and those where selection makes a strong contribution, mutations on that borderline are common in protein evolution, was her original formulation. And so the idea is that fitness effects may be nearly neutral, but in fact the evolutionary dynamics for such mutations are quite different from strictly neutral ones. In both the rates of evolution are strongly dependent on population size, and adaptive evolution can be quite prominent. And so the motivation for this model, one of the motivations was rates of protein evolution that differ, that seem to be difficult to reconcile for strictly neutral models. And so one of the patterns was faster rates of protein evolution in primates than in rodents. And this is a recent large-scale study, but that basic difference in rates was seen even in the 1970s from initial vertebrate sequences. And so here, this shows the average protein evolution scaled to synonymous divergence. So that should adjust for differences in generation time and mutation rate in these lineages. And yet we see a twofold difference in the average rate of protein evolution. And so Ota's argument was that these borderline mutations, right? So here we have a schematic where new mutations enter the population. If we take sort of a simplified view of directional selection, we can consider that a bias towards reduction or increase in gene frequencies, and then drift is a random force. So in Ota's view, these forces are of roughly equal magnitude. And of course, the extent of drift is highly sensitive to population size. And so her argument was that mutations that had sufficient deleterious effects to be eliminated from rodent populations could drift to high frequency and go to fixation in lineages, in primate lineages or ancestral primate lineages. And that could cause the rate variation we detect. In fact, she would have to argue that at least half then of the amino acid changes that have occurred in primates have been deleterious. And so it's arguable, yes, but that's her explanation. And in fact, there's evidence that this kind of borderline mutation is important in protein evolution, and the evidence comes from rate variation, faster rates of evolution on islands or in regions of low recombination, and also population genetic evidence that we tend to see non-synonymous changes as rare polymorphisms compared to synonymous changes. And that's true in protein evolution, also within regulatory regions, and we see that at synonymous sites. So this seems to be a prevalent fitness range, both among functional categories of mutations and in different taxa. And so the prevalence is also quite curious, because probably one of the weaknesses of the model is that it only works in a very narrow range of parameters. But my goal was to try to develop a strong case for weak selection, and the choice of system was synonymous mutations. And so I'll first motivate this model of major codon preference that's been discussed already in this session, and then present a test, a population genetic test of the model, and then move to some of the consequences that we might observe, that is, strong fluctuations in codon usage evolution among lineages. And part of the reason for looking at this is to determine how we can use synonymous sites to study protein evolution if they're not neutral. Right? It's clear and it's quite useful to have some neutral sites to calibrate protein evolution, but they can still be useful under near neutrality, I think. So some of the cleanest sort of genome-wide patterns I've seen come from plots of codon usage versus estimates of gene expression. And these data come from yeast, where the expression estimates are transcript abundance. And what I show here is a particular synonymous family, lysine, which is encoded by 2 codons. So A to G mutations in the 3rd position are synonymous. And here, AAG is a major codon, and that's identified through data that's independent of this relationship, mostly from experiments by Ikemura that revealed high abundance of tRNAs that recognize the AAG codon. And there are similar patterns for other synonymous families. And so I show some examples for different redundancy, so twofold, fourfold, and sixfold families. And importantly, in yeast, different nucleotides can be favored at the third position. So we have T and A favored in some cases, and C and G in others. And so it's possible, and there's some support for either mutation bias differences or gene conversion differences depending on transcription rates. But here, one would have to also argue for very strong neighboring base effects, right, to argue that these could be purely mutationally driven. And in all these cases, the red codons come from the Ikemura studies independently of these patterns. And so that's all in Saccharomyces cerevisiae, but I'll be looking at data in Drosophila. Right. So those are just genome patterns. How does this relate to what we know about the process of translation? So this shows some ribosomes programmed with messenger RNAs. So at each codon, there's two important aspects. One is the waiting time for the arrival of a tRNA, and then once the tRNA has arrived, there's some processing on the codon, on the ribosome, and the kinetics of that is quite well known. There's at least 2 stages of proofreading where non-cognates and near-cognates can be rejected. Right? And if a codon or a tRNA isoacceptor is accepted, then the peptide bond is formed and the next codon can be translated. And so the cellular abundance should clearly affect the waiting time during protein synthesis, and particular aspects of the codon-anticodon interaction might also affect the subsequent processing, right? So some codons might have higher rates of cognate acceptance given their arrival, and importantly, high rates of distinction between that codon and non-cognate codons. Right? And so there are several ways that natural selection might discriminate among codons at the level of translation. And basically, the notion is that major codons should allow faster elongation and more accurate synthesis at each step. And so the fitness consequence should be that an organism can more efficiently use its pool of ribosomes, that ribosomes return more quickly to the free ribosome pool. So it's thought that for many proteins, the rate of protein synthesis for a given gene might be more determined by the initiation rate rather than the elongation rate, but the fitness advantage might be more the global use of ribosomes. Right? Although increasing accuracy should reduce the required number of initiations also for a gene. So the idea is, especially for organisms that might be fast-growing or have limited resources, that codon usage bias would allow faster and more efficient growth. And that this is a co-adaptation between the tRNA pools and the codon usage in DNA. So in some ways, when I was starting these studies, there were some papers from molecular biologists that seemed to accept this model. But it was a little harder for population geneticists, at least the people that I was talking to believe that a single synonymous difference between, let's say, 2 Drosophila individuals would be detectable. Right? That would have a sufficient effect to be discriminated by selection. Right? So in some ways, my motivation was to provide evidence for population geneticists. And so an appealing aspect of the codon usage is that we can capture some of the main ideas in a pretty simple model, at least to use as a starting point. And what's been surprising is how many aspects of the data seem to fit pretty nicely to some predictions of the simple model. So here we have a model of two states, two possible states, a major codon with a fitness advantage compared to its minor counterpart, and then possibility for different forward and backward mutation rates. And so a nice feature that comes out of that model is that silent mutations fall into fitness classes, so slightly deleterious mutations from major codons to minor codons, and advantageous mutations in the opposite direction. So that seems obvious, but that's not something you can do for proteins, or it's not something you can do very readily. And it can be extremely convenient because you have classes of mutations where the evolutionary dynamics or the patterns can be compared. And another aspect of the model is that although selection coefficients might be strongly dependent on the number of translation events at a given codon or the expression level of that gene, the basic model should be the same. So although we might have to take some care, it's possible to pool or analyze data across a large number of genes. So this is a model that should be broadly applicable. And so that should give some statistical power to our analysis, especially since we're trying to detect very small fitness effects. And so Lee and Bulmer have studied codon bias models, and so one can calculate the rates of per gene substitutions in the forward and backward directions. If we set them equal, then we can find an expression for codon bias, given that these parameters have remained constant for a sufficient period of time. So basically, this is the level of codon bias that can be achieved if this model is affecting a gene, let's say made up of AAG codons, and the parameters hold across all the codons. So under that model, we can see the codon bias variation can be due to either differences in mutation biases, and so we can tell for the zero selection case. I chose 3 mutation biases that were spread out somewhat here. And then, of course, selection coefficients or population size can affect the codon bias. And so what's important here is that selection, in fact, if this model holds, selection has to be weak, right? Because what we see is intermediate levels of codon bias, and we rarely see 100% major codons, and at this level there would be no synonymous divergence. But synonymous divergence for highly expressed genes is suppressed, but only a 5-fold or so compared to what we think the neutral values are. Right? So if this notion is correct, then we really, the selection coefficients should be in this range. Right? And we can see that the codon bias is quite sensitive to changes in NS in this range between or close to 1, NS close to 1. And so we have a system where we can, if we can infer ancestral states in a data set, then we can assign the fitness classes to synonymous mutations. And so basically we can look at within and between species sequence data. And so the question is, well, should we expect to see some signal of weak selection? Right? And we have a pretty good idea of how weak that selection should be. And so one aspect of the data we can look at is basically the locations of mutations on the gene tree. Right, so this is an example for neutral evolution, and one aspect here is the level of expected divergence, of course, depends on where the neighboring species is. And so this is taken to fit data for Drosophila, melanogaster, and simulans. And so what's plotted here is kind of a summary histogram for the locations of the mutations. So the mutations that are found on only one of the alleles from within a population, these two and these three are at frequency 1 in the sample, and so about 30% of the sample we expect. Mutations that have occurred before a coalescent, here this is at frequency 2, this is at frequency 4, and then the frequency 5 class has the subscript F. These are the ones that are fixed, at least in this sample. And then the theory predicts also then how this, how the locations of the mutations and then the histograms should shift with selection. And so we could see even for fairly weak selection, and this was adjusted to give codon bias of about 80% major codons, which is what we see in many of the genes that we examined in Drosophila. So there is some shift towards deeper locations, more fixed differences, and higher frequencies for the preferred mutations, in contrast to the shift towards rare alleles and lower substitutions compared to the neutral model for the slightly deleterious mutations. Right. And we can also, under this idea, if we have some estimates or plausible values for mutation rates and selection coefficients and divergence times, we can simulate under our specific assumptions, we can simulate the numbers of sites in a sample and test the statistical power, right? So the key aspect is is weak selection detectable in at least when recombination is pretty frequent. And so these graphs show the statistical power, that is the probability of rejecting the null hypothesis, which in this case is equivalent distributions of frequency distributions and divergence for our two putative fitness classes. So the probability of rejecting the equivalent distributions under varying degrees of codon bias or varying degrees of selection in this case. The mutation bias is held constant. And this shows statistical power and its dependence on the numbers of alleles and codons that are sampled. And there are three graphs. So these show, if we look at the full histogram, both the frequency distributions of mutations and the divergence compared to tests of only polymorphism versus divergence. So this pools all the polymorphic classes. And so this is a 2-by-2 comparison. And this is a comparison that only looks at the polymorphisms. And so this shows the gain in statistical power by using more of the available information. And we can also see, so for the same increase in the total numbers of codons analyzed, that a 5-fold increase in the number of alleles has a smaller impact than a similar increase in the number of sites. Right? And so this was important because the available data, we had a large number of genes, but a small number of alleles. So this was comforting. This meant that we should have a high probability of seeing a signal of weak selection if it was there. Right? Well, to be honest, this was done after we looked. But so this is, again, the expected pattern for selection sufficient to give about 80% preferred codons, which is what we observed among 40 genes or so in Drosophila simulans. And the difference in the distributions was strongly significant, and in the direction that was predicted, that is higher fixations and higher abundant alleles for preferred mutations, and excess rares and decreased fixations for unpreferred. And what was remarkable was how close these distributions are to the expected distributions, because this is a pretty specific and in some ways unrealistic model, especially the notion of completely free recombination and statistical equilibrium for the polymorphism data. So this was maybe somewhat of a surprise. And so we can take data like this and estimate the best fit NS values, and they do come close to what we'd expect for weak selection. Right? And since then, there have been a number of models that relax some of those strong assumptions. I think Brian might have talked about some of those more sophisticated estimates using similar data. And I think the NS values come out to be pretty similar in the species that we looked at and also in some other species. Right. I'll show that in a second. Yeah, why not melanogaster? Well, so yeah, it turns out melanogaster has. Oh yeah, so here it was which species had sufficient data was the major criteria, and really besides melanogaster and simulans, there wasn't much choice. Partly because we need closely related outgroups to do this analysis. So it's not just a matter of having polymorphism data from within the species. But there is a reason why this is not melanogaster that I'm showing. Right. And so I would argue then that this provides some evidence that the codon usage, at least in Drosophila, is a good candidate for OTA's kind of weak selection mode. And that is that, as far as we can tell, the preferred changes provide some fitness advantage, and that the fitness advantage is sufficiently small that minor codons persist by genetic drift. And so some of the consequences of this would be that that intrinsic to this model, even at equilibrium, is that half of the substitutions that occur are adaptive. That is, because deleterious mutations can drift to high frequency and go to fixation, that adaptive fixations are required even to maintain a given level of codon bias. Right? So under weak selection, if you think of Kavita's climber climbing the adaptive peak, he'd actually be slowly sliding down, right? Because the fitness effects of the deleterious mutations are sufficiently small. So just to maintain his place on the mountain requires some climbing. And then secondarily, an important difference with the neutral model is that we might see very different evolutionary patterns, even with small changes in population size or changes in selection coefficients. And so the previous model showed this is a stationary model or equilibrium codon bias, but you can imagine even a twofold shift, let's say from 0.25 to 5, is quite a jump in levels of codon bias. And we can see that better here. This shows if we have some initial level of codon bias and allow population size to change, either a twofold increase or a decrease to 1/3 of the initial value, then we see a change in the new equilibrium, and the achievement of the new equilibrium is quite slow. It's of the order of reciprocal of mutation rate. Right. And so this would suggest, depending on the time scale of fluctuations, right, the actual codon bias we see in nature may often be out of equilibrium. Right? And so that's a prediction of the model, and that's also something that we can test. So similarly, we can assign the ancestral states and simply count and compare the numbers of preferred and unpreferred mutations. So we expect roughly equal numbers at equilibrium, and small changes in parameters can give fairly substantial departures towards excess preferred and unpreferred. And this might be a way that we can also study protein evolution. That is, if we see fluctuations in codon bias, if we can attribute them to changes in effective population size, for example, then we can test for the types of protein changes and the numbers of protein changes that occur in lineages undergoing adaptive synonymous evolution or deleterious synonymous evolution. Right? And particularly if protein evolution is largely adaptive, we might see patterns like this. So that's the motivation then to try to look at more lineages, now not just polymorphism and divergence within a lineage. And of course, we have to pay attention to this ancestral inference method and how reliably this can be done. And that turns out to be a little bit more tricky than I expected, and I'll show some data about that. So we're looking at species in the Drosophila melanogaster subgroup, mostly because there was sufficient data at the beginning to do polymorphism studies in melanogaster and simulants. But it turns out there's a nice topology in that most of these branches are sufficiently long that we should be able to see interesting non-equilibrium patterns, but the species are sufficiently closely related that we have some reliability in ancestral inference. And so this split, so that one just might be too close to really, but I'll show, I'll show some data for Shisheliya. Yeah. Mauritiana might be too similar to simulans. Yeah. But the deepest split here is shallower than the human mouse split, at least for synonymous divergence. So this is fairly closely related species. And they have quite different distributions, although I'm not sure that would necessarily indicate differences in population size. But two of the species, Drosophila orina, was isolated, collected only once on a volcanic mountain in Cameroon and never found again, even though there were searches. And then Drosophila Shisheliya is an island endemic that is also a specialist. Right, and that's compared to several species that have much wider distributions. And so we did a study of about 20 genes where we compared sequences in 6 of the species in the melanogaster group. And we avoided very close relatives of these species just because the lineages might not be long enough. We couldn't count sufficient numbers of changes to assess departures from equilibrium. And one aspect that was a little bit of a surprise at the beginning was that Drosophila melanogaster has a uniform and quite large excess of unpreferred changes, right? So these aren't necessarily all fixations, because some of these genes we don't have polymorphism data, so we're not assigning pre and post most recent common ancestor changes. But in cases where we do have that information, this pattern seems to hold. That is, the numbers of unpreferred mutations are higher, consistently higher than preferred. Right? And the overall bias is pretty strong. And these are ranked in order of the skew, how much bias there is towards unpreferred mutations. So that's why I didn't show the polymorphism data in melanogaster. But that sort of pattern, loss of GC, has been seen in several mammal lineages and other Drosophila lineages. For a reason I don't understand, we very rarely see GC increases, at least when looking at closely related species. So we were glad to see at least one of our lineages seem to have a predominantly preferred substitutions, or again, changes in the lineages. We don't know if they're actually substitutions. And there are a few genes that have similar numbers of preferred and unpreferred changes, but the vast majority, and in some cases, that excess is quite strong. Right. So, but in both cases, 20 genes, we can say it looks like a global pattern, but we can't say for sure. And more importantly, it's difficult to test for the underlying cause. Of course, a change in mutational bias can give us these patterns, right? And we're sort of more interested in seeing changes in NS, partly to see what happens to protein evolution. So we were fortunate that now there are 4 outgroups to melanogaster where genome sequences are available. So Shashelia was not in our previous study, but we can do analysis now. And then currently, we're trying to get some genome-wide data for TCRI and Orina. But with currently available data, we can look at least six lineages, although these get to be a little bit long for ancestral inference. Right? Well, so the simple model, right, predicts how we can identify selection at equilibrium. It also gives us a way to test for the causes of lineage-specific fluctuations. And so what this shows is in a case where population size has decreased to 1/3 an initial value, and that decrease is the same for all the genes in that genome. Right? But the genes start with different initial levels of codon bias. And so that could be due to differences in expression level. For example, there's variation in NS, but all the NSs decrease to 1/3. And this is the instantaneous change in codon bias, and the statistic is a skew statistic. It's simply the percentage of preferred changes minus the percentage of unpreferred changes. And we can see for that NS decrease. And usually, it's easiest to think of a population size decrease, and that's a pretty modest decrease, probably. But it's also possible that translational selection is more strong. Fitness effects related to translation are stronger in some taxa than in others. For example, if growth rate is more important determinant of fitness. Right? But the assumption here is simply that it's a uniform change. And so what we see is, of course, no change if selection's not acting. And for codon, genes with intermediate codons, we see a fairly strong decline in codon bias, so some skew towards the excess of unpreferred. And for genes that start out with very high bias, we can see a much stronger pattern. So this change in NS causes a great deal of differences among genes in the change in codon bias. And similarly, for an increase in codon bias, again, if that NS change is uniform for the genome, we see something almost linear in both cases, but genes that have initially low codon bias should see little change, and high bias genes should show the bigger departure. Right? And then in contrast, of course, for the mutation change, the substitution rates are just directly proportional to the mutation, right? So we just see a uniform change. right, across genes. So it seems that we should have a method then to identify in cases, and maybe it's simplistic to think that it's just a uniform change in NS. But that could happen for changes in population size, for example. And the uniformity is testable. So one issue is this ancestral inference. And I'll just briefly show, we did some simulations. So this is what we think the distances are in the melanogaster subgroup to do sort of a stress test on the ancestral inference. We tried doubling all the lineage lengths, right? So it's a little bit more of a challenge for ancestral inference. And so parsimony is sort of the simplest method, and this is a known bias. So when the character states are unequal, parsimony overestimates the changes towards the less common character, right? And it can be a pretty strong bias. So this is the real. This is a simulation of equilibrium codon bias. But what we get is a pattern that's strongly different and to us looks exactly like what you'd expect if NS has decreased, right? So there's a real concern about ancestral state inference. And so you might try a more sophisticated model, a maximum likelihood model. But interestingly, for this model, which allows for base composition skews, we still see a bias, not as strong as parsimony. And it turns out that's because it's not an exact fit to how we simulated. And what was even more disturbing was a non-stationary version of this likelihood model did even worse, right? And that seems to be because the substitution model is incorrect. Adding non-stationarity took it even further from the truth, even though that was some realism that we were trying to add. And so now we've convinced Ziheng Yang. So he's developing all these inference methods. So a more sophisticated general time reversible likelihood model did a better job. Or we were able to simulate that model correctly. So it's kind of instructive. We were doing the simulations, whereas Yang was doing the inference methods, and it was hard for us to get the right model. So you can imagine it's pretty unsafe to assume that these are the correct models in nature, right? But we think at least we have something that should capture a lot of the aspects of the data. But this is just a kind of a warning that this is something we have to be careful about, even in the closely related species. Although this might be slightly exaggerated, right, compared to the actual distances we're looking at. But having said that, I'll just give one example. This is Drosophila shishellea, and this is the data summarized. So genes are binned by their initial codon bias, and we can see that genes of low bias ancestry in that lineage have little change in codon bias. High bias genes have had a big decline, and there's actually a very nice fit to a linear model. And this, of course, is an island endemic species, so it might be reasonable to think that the population size has been smaller. But we can get this fit from just a small shift in the population size. I think if you ask most Drosophila biologists, they would think that the population size is much smaller than a third of melanogaster and simulans, and we don't need to invoke any change in the mutation bias. And so this is just one of the lineages. I'm not going to show the other ones, partly because we're still we want to do a little bit more testing with the inference methods. But this is the hope. And so both the population genetic analysis and the lineage analysis, we've been able to take maybe an unrealistic simple model, but the fit of the data is quite good. And so I hope to have shown that we can develop some powerful tests, even of weak selection, and we can confirm some of the expected consequence of that kind of selection. And one of our goals is to look at protein evolution and see if we can use similar methods to detect potentially similar kinds of selection. That is, if natural selection is discriminating among synonymous codons, the same translational selection one would expect to also act among non-synonymous codons. Right? Some of the selection for codon bias is between non-overlapping or tRNAs with non-overlapping sets of cognate codons. That's exactly what non-synonymous codons are as well. So of course it's complicated because there are other constraints on protein evolution. Right, and I have some data about amino acid composition, but maybe I should stop there for time. A little polymorphism in Shashelia. Right. 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 Mm-hmm. Right. 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 So this curve assumes equilibrium, codon bias, and it's the instantaneous change for a change. So we know that's not true. Yeah. So yeah, you're right. If we think of but we don't know if we should consider equilibrium population with polymorphism, for example, and then allow a branch with a small population size. Yeah, you're right. That's something we need to look at. Yeah. And of course, a real interest is what does melanogaster look like? Right. Yeah. Right. Right. Well, so in yeast, the tRNA copy number is a pretty good predictor of tRNA abundance. And so I think we're hoping that the same thing is true because it's a lot easier to count tRNA copies in eukaryotes, right? Then very few people are measuring tRNA abundances anymore. But the question is more about tRNA abundances rather than gene copy number. The evolution of tRNA abundance. Yeah, yeah. So we're assuming, here we're assuming the major codons identified in melanogaster actually are conserved, but we're finding that melanogaster has a lot of evolution away from those. But most of those patterns, right? These not much evolution has occurred on these lineages. Most of those should be ancestral, shared ancestral codons, right? But yes, so tRNAs could be shifting among species. The preference might shift. So there's a possibility that you could actually change the major codon. Oh, right. So that's a possibility for the melanogaster pattern. And in fact, as Brian alluded to, there's been an argument that there's been a change in preference in the melanogaster lineage, right? So the AT codons are generally preferred. So I find that a little bit hard to imagine, especially because your highly expressed genes, your ribosomes and glycolytic enzymes, are adjusted or co-adapted with the tRNA pool. So if you shift the tRNA abundances, now all those genes have very low translatability, right? So there's also notions that there can be tissue-specific codon preferences. But I also find that a little bit hard to imagine because the housekeeping genes, and a lot of them are single copy. So I suppose if you had tissue-specific housekeeping genes, and then you could see the codon bias differences between those, then it would be a better argument. And in the one case where there is a strong tissue-specific codon bias, but this is among non-synonymous families, so in the silk glands of silkworm moth, what happens is a very small number of cells produce a huge amount of silk protein, and the tRNAs before silk production are adjusted, and it's a highly repetitive protein, silk. And you see a nice shift in tRNAs just prior to this high silk production. But interestingly, those cells synthesize ribosomes and housekeeping proteins before shifting to silk production. Right? So there's kind of a temporal way to avoid the problem. So I would think that, and Ballmer's model of shifts in codon preferences is a loss of codon bias, right? And so that would be, low NS can put you back at mutational equilibrium, and then you re-evolve it to another state, right? And mutation biases can influence that evolution. There's some interesting possibilities, though, where there could be some favoring. So, for example, some RNA viruses prime off tRNAs. And so you might think, well, those viruses might adapt toward abundant tRNAs and then cause pressure on the hosts, right? That would be interesting. I think yeast has several cases of this kind of RNA virus. And we do see, if you compare very distant species, yeast and E. coli have some clearly different codon preferences for some synonymous families. But other synonymous families are pretty consistent across very distant taxa. And so I mentioned that the abundances should affect the arrival time, right? But this processing on the ribosome, there might be some aspects of particular codon-anticodon interactions that allow faster recognition and faster rejection of non-cognate. So there might be some intrinsic aspects of particular tRNAs that allow them to be translationally advantageous. Yeah. And there's a few groups studying kinetics of protein synthesis. So there may be information about that.
So remember these old experiments where I think it was Carlini or something who modified the ADH synonymous sites in Drosophila larvae, right, and found pretty big effects on the phenotype. So I'm wondering whether part of why you're finding weak selection, and in the short term you do find, even in sort of similar species, very strong selection, the potential for it, maybe there's compensatory mutations even within the short time frame of the lineages that you're looking at. And therefore, I mean, I don't know if this is true. I'm just wondering if this is true. If that leads to it appearing as if, they're under very weak selection, whereas they may start off being, I don't know, just make out what's going on.
So I believe your name is pronounced Agashi? Yeah. So so this is what I've been thinking of for the last couple hours. So there are some differences, I think, between our findings because different selection pressures in prokaryotes. So, for example, the Shine-Dalgarno sequence avoidance seems to be a pretty important factor in E. coli, but that shouldn't be a factor for eukaryotes. But you mentioned there is some experimental evidence for Drosophila, so this won't hold for my argument then. But so the contrast is between experimental and comparative or population genetic differences, and important consideration might be that so all my analysis looks at evolving sites, polymorphic or fixed differences. So if strong selection is acting and is consistent, then we might never see those mutations. They never make it to my analysis. Whereas in at least some of your experiments, the reversion selection, it detects your filtering for strong effects, right? So I would think, so that means there are some, and also in eukaryotes, some synonymous changes that can have strong effects. And we just have approaches, the experimental methods are biased towards finding those, and the population genetic methods might be strongly biased towards leaving those out, right? And then that's why Brian was asking about whether you looked at polymorphic sites. And I think the Carlini study, so this is a study in Drosophila, a replacement of preferred to unpreferred mutations, they chose some of the synonymous changes that seem to be they chose some of the rarest codons, leucine, in Drosophila. And so I think if you did this similar comparison, look between species, those might also be the most conserved. And leucine in yeast, I think some of those codons are also quite prone to frame shifts. And so there could be something particular about those. So sure, I guess the way to resolve this would be to take some of the codons that vary within populations or between species and try the experiment in those and see if you can get strong effects.
I think there's a kind of what we call an elephant in the room in Britain, which is, I think the evidence for a relationship between codon usage bias and effective population size is quite weak. And so, for example, in Drosophila melanogaster, if you compare codon usage bias across regions of the genome with different recombination rates and different levels of genetic diversity, there's essentially no relationship till we get down to the very low recombination parts of the genome, right? And again, the codon usage bias in lots of organisms is not that different apart from mammals, which seem to be peculiar. So I proposed last year that there's an alternative model which is stabilizing selection, and that under that model, when you calculate the relationship between NES at the level of individual variants, it's almost independent of NE over a wide range of NE values, which but it's kind of indistinguishable from the Lee-Bulmer model at the level of the behavior of individual variants.
Right.
And I wonder if you have any thoughts on that.
Right. So, so the stabilizing selection model, there's sort of an optimum codon bias level.
Yeah.
Yeah. But then if you're at the optimum, then both the preferred and the unpreferred changes should be deleterious.
No, because of mutational bias.
Right. Oh, no,
That
right.
drives
okay.
you slightly away from the optimum.
But
and that's
then you
sufficient
get
to give
the
you
same,
a directional selection
right.
component.
Then even if it's stabilizing selection,
Yeah.
then effectively it's
Yeah.
the same model here. Right.
Yeah, it's effectively the same
Right.
model, but quantitatively it behaves in quite a different way because it creates an independence between NES and NE.
Right. But so then, so that is a major problem for this model, but then the stabilizing selection model, so a highly expressed, so for some of the yeast codons, you do go close to 100% for the highly expressed genes. So the stabilizing selection model would, how do you account for the among gene variation?
Well, that is a problem, I freely admit. I went through some convolutions in my paper to try and solve it, but I'm not sure I'm totally convinced by my own convolutions. But, there seem to be problems with both models.
Right.
And so maybe something else is going on completely.
Right. Well, so I guess the other maybe escape explanation is that the codon bias levels are very long-term products, whereas recombination might be fluctuating more in recent times. So the levels of variation are correlated, but that's a much faster response to recombination rate.
That's certainly a possibility. We're hoping to try and test that by applying some of the population genetics models to sort of genome-wide polymorphism data and see whether or not the estimates of NES vary according to recombination rate. But whether that'll work out, I don't know.
So that would be right. So in the long term, I think this is sort of Bulmer's model for how you shift the codon preference. But here, if we just look at the substitutions that occurred, there's a huge excess of unpreferred. But in the genes, it's still 80–90% preferred codons. Right. So I don't think that until you really accumulate a lot of unpreferred codons, you don't expect a selective shift in the tRNAs.